for the benefit of those that were not here last week, this is our agenda today. We're going to start first by recapping transcription and translation. And after that, we will go into mutations, which is what I need your class activity for. Then we will clear any doubts you have. Then I'll distribute RE 6.1 due on the 26th of August, which is like 10 days from now. And I hope you have some learning device, either iPad, worst case is your phone also can work. But later on we're going to do an activity. Here we're going to mutate some stuff. Okay, you're done. Those that didn't come, you do the activity 1, the crossword puzzle. Anything you would like to check? Yes.
8 downwards.
Okay, 8 downwards. What's the statement?
Where does translation take place?
Where does translation take place? What do you write?
Ribosomes.
Oh, it takes place at the ribosomes. And it fits in now.
Okay, I know what it is.
Oh, okay, then what is it?
Cytoplasm.
Oh, okay. Okay, so where does it take place at? Anything else? Number 2 across, some students write complementary. Yeah, it would help, it would fit, but it's not complementary. It is RNA polymerase. Okay, yeah. Okay, 5 down. First amino acid that will be translated, is it always methionine? Yes. Okay, so we'll start up this way. To get us all on the same page, I will play a video that shows transcription and translation. So you know the broad picture. Then we will go down into the mechanics of it, any areas you'd like to clarify, and then we will clear everything before we mutate the DNA. Okay, so along the way, last week I got your classmates to join in at, let me pause this. Okay, as a QR code, I'd like you to work with. Okay, so I'll leave this Q&A portal on the entire lesson. Along the way, you have any questions, but you feel like you don't want to interrupt or anything, you just put your questions up there. Then we'll clear it along the way. Okay. So before I play the video on transcription translation, you could scan this QR code. If you have questions along the way, I'll be glad to try my best to answer. If not, we'll find out the answer together. Is it what? Is it not? Okay. Okay. If not, right, just shoot the answer straight in my face also can. That'll be better. Okay, this is to capture any straddler questions that I didn't get to answer. Okay, let's go. Okay, so a quick video to recap to put us all on the same page.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called a nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Alright, okay, so that was a quick recap of transcription and translation. I'll now take some questions from the floor. Okay, any? Yeah, I'll pose one question to you first. Where does RNA polymerase attach to begin its function? Okay, if your friends are posting questions, you see anything you like, you can also upvote it. So if you have a question, just ask me along the way, okay? Right. Then my first question to you is this. Where does RNA polymerase attach to? This is a gene. Okay, this is a gene. Where does RNA polymerase attach to carry out transcription? Okay. Do you see here? Yeah, so where does RNA polymerase attach to carry out transcription? It is the start codon. You know, okay, so it's not the start codon. Okay. Okay, it's a promoter. Okay, very good. Yeah, okay, you all need to give me a more shocking response. Okay, so it's a promoter. Okay, the promoter is upstream of the gene. So the promoter will be somewhere here. RNA polymerase will bind to the promoter. Once it recognizes it, then it will begin its transcription. Okay. Next question. What is the promoter? Okay. The promoter is actually a sequence of DNA. It's not protein, it's not fats. It is a sequence of DNA. The reason why we can't just write promoter is because we don't need you to know that sequence of DNA. However, there is a specific sequence. I cannot remember the sequence, but I think okay, I'm just writing up. I know it starts something like that. But a promoter is just a sequence of DNA. It doesn't code for anything. It is not going to result in any amino acids. It's just for recognition by the RNA polymerase. So last time when I was in university, when I wanted to find a gene, I would go and look for this code. I'll type this code into the machine, and I'll ask for it to search for this code. If it finds the code, it means that there's a gene next to it. Yeah? Okay, so this is a promoter. Yeah, in general, the promoter is the same. Yeah? So there may be some differences here and there, but this is in general the code that we will type in if we want to find a gene. Okay. RNA polymerase has two functions. The first function is that after it attaches, it will then unwind the DNA. Yeah, it will first unwind the DNA. Make it available for transcription. Okay, so I've unwound the DNA. Now, which strand do I use for transcription? That's the next question for you. Which strand do I use for transcription? There are two strands actually. Yeah, which strand should I use? This is for the benefit of those that are not here. Okay, which strand should I use? Top or bottom? Okay, who says top? Bottom. Okay, but why bottom? Okay, so I see that you have identified ends. So here's the 5 prime, here's the 3 prime, here's the 5 prime, here's the 3 prime. We read the template strand in the 3 to 5 direction, and we build in the 5 to 3. So what does it mean to build in the 5 to 3? This is the first one. Okay, let's build up. If this is the 5 end and this is the 3 end, if I'm building in the 5 to 3 direction, it means I'm adding new ones to the 3 end. I'm adding new nucleotides to the 3 prime OH end. I'm building it in the 5 to 3 direction. So I build my mRNA in the 5 to 3 direction, but I read what we call the template strand in the 3 to 5 direction. Okay, this is the template strand. Let me just build the entire thing first. G, C, and then we put a A. Okay, questions? I have a question for you. If we say that we build mRNA in a 5 to 3 direction, then what's stopping me from doing this instead? Why don't I build my mRNA again? It's also 5 to 3 one. Yeah, I'm going to go the other direction. So, if I add one more term, right? I'm building in a 5 to 3, right? I'm building the mRNA in a 5 to 3. mRNA has a 5 end, it's a 3 end. I'm building in a 5 to 3 end. What if I elongate this and use the top one as a template? Why not? What about the promoter? Okay, so it has to do the promoter, right? I'm using this bottom strand as a template strand because my promoter is on this side. If I wanted the top strand to be the promoter would be found on this side, and it would be, the sequence would be, what? Promoter. The sequence will be the other way around. Yeah? So, in reality, this is not something that will be explicitly told to you. When I was your age, I had the assumption that the entire DNA strand, only one of the strands, right, will contain all the meaningful code, the templates. But it's not true. When I went to university, I started typing the promoters, right? And I got like, hey, what am I seeing? I see some promoters this way, then I see some upside down, then I see some upright, then I see upside down. Then I realized actually both strands can be the template. Yeah? It just depends on where you find the promoter. The promoter is this way, the bottom one will be the template. The promoter is found the other way, top one will be the template. And so we find promoters arranged differently across the entire DNA sequence. Okay? But this is not explicitly said in the textbook at all. Just for your information. Last week, Megan also said something interesting. If we say that one other way to see is that the mRNA that's produced should have a start codon embedded at the very beginning. Yeah? If we it does not, then something is wrong. Okay? So AUG, another universal sequence that you can find across all the mRNAs. It begins with AUG, that's the start codon. That's what the ribosomes will bind to. Okay, and recognize as the first thing to translate. You know the vaccines we're taking now? We are injecting mRNA into us. Yeah? And when this mRNA goes, it will be taken by our ribosomes to translate protein that are viral in nature. So essentially we are throwing in recipes to create a virus, parts of a virus within ourselves. Then our ribosomes will synthesize parts of the virus. But it's not harmful in the sense that these viruses aren't complete. Only parts of it, maybe a limb or two. Yeah? So all it triggers is the immune response without hurting us in the process. Yeah? Okay, so this strand, what we call the template strand, the top strand often is referred to as the non-template strand. Okay, but sometimes it's called the coding strand. Does it have any value? Right, we call it the coding strand. Okay. Personally, I like calling the top strand a coding strand because it has meaning when we say it's a coding strand. When we look at your mRNA, you see that actually information is coded there in triplets. AUG, GCC, AUG, GCC. I'd like you to try to read the genetic code now. I'd like you to read a translator. Okay, on page 55 of your textbook, you will see a table filled with codons that translate and tell you what amino acid should be put in a particular sequence. Based on this sequence you see here, what amino acids will you get? Okay, so a quick practice. Okay, how many codons are there? 4 codons. Okay, so what's the first amino acid? Methionine. Second one? What? Lime. Okay. Glide. The next one? Van. Next one? So what are you reading? GCC. Ala. Okay, so it's ala. Then after that? Ala. Oh, methionine. Then after that, ala. Hey, you are really right, man. Ala. Okay? Yeah? Good. Stop codon. My mRNA is a really short one. I did not print anymore. It should have a stop codon. So by right, we should have three more sequences here that will result in maybe a UAG. I think that's a stop codon. Okay? So there needs to be a start and a stop. Okay. So we call this top strand a non-template strand, also the coding strand, because if you notice the sequence is quite similar. ATG, AUG, GCC, GCC, ATG, AUG, GCC, GCC. The only difference is that the T is above for the U. So that means to say, if you are ever given the coding strand and no mRNA given, you can also use the genetic code to read it. Okay? The genetic code can let you read two things: the coding strand and the mRNA. The template strand is not the thing you do not want to read. Right? Reading the template strand will yield you different amino acids altogether. How will you check if you are reading the correct thing? Well, the protein should always start with methionine. So if you go and read TAC, you shouldn't get methionine, if I'm not wrong. Yeah? So that's one way you can check. Okay? Last week, I asked the class, half of the class at least. Okay? Often, when we teach this, one of the things we say is this, that transcription is a photocopying process of the DNA. Can I say again? Transcription is a photocopying process of the DNA, double-stranded DNA. To what extent is this true or false? Right, 5 marks, right? They got level 1, 2, 3. So, to what extent is this true or false? Transcription is a photocopying process of the DNA. To what extent is this true?
True, but not very true.
But not very true. Who was not here last week? Alright. Then I'll give you a thing. Sandy, what do you think? To what extent is this true or false? To a certain extent. To a certain extent it is true. Okay. What element of my statement is true? That transcription is a photocopying process. Okay. Other than the T replaced with a U, the sequence is the same. Okay? So therefore it's kind of like a photocopying process. Okay? So that's to a certain extent. Right? How will you counter my statement? Okay, what's not exactly the same? This strand. mRNA. Okay, the mRNA is not exactly the same as the coding strand. Okay. Anything else? Okay. If it's a photocopying mechanism, we should refer to this to create this. But actually, we need to refer to the complementary sequence to generate the mRNA. So it's like a complementary photocopy. Right? We make use of the template to create the mRNA, not even the coding strand. Right? A complementary photocopy. But it's not a photocopy because it's like U but not T. Okay. Anything else that makes it not a photocopy? Dominic said, oh, you watched the video. Dominic says it's shorter. Yeah? Dominic's not here, but his words are always with us. Okay? The mRNA is shorter than the DNA. Right? So it's not exactly a photocopy. Okay, so but there is a process in our body that photocopies DNA. And that process is DNA replication. So I can say that DNA can be photocopied. That process is DNA replication, but that process is not transcription. Okay? Okay, so let us now answer some of the questions posed by your friends. Number one, what are the purpose of introns? Okay, there's one page of your textbook where they mention introns and exons. What page is that? 52. Okay, 52. Introns, exons, for your information only. Okay, so you see the word splicing. That's all for your information. Don't need you to really know this in depth. But let's try to make sense of what introns and exons are. Okay, in reality, the mRNA that is transcribed is not all useful. There's quite a lot of not useful stuff within the mRNA. For example, let's say I've transcribed my mRNA. Okay, I'm going to color code it up. Some segments are blue, some red, some blue, some red, and some blue. Wow, I can't remember. Introns are the ones you keep, exons are the ones you take out. Exons you keep, introns you take out. Okay. So let's say I'm going to label the red ones as introns. Exons. Okay, mind you, this is a mRNA. In reality, the mRNA that we transcribe is not all useful yet. There's some not useful code in between. Yeah. What our body does is that there will be a little bit of processing. Okay, the word is processing. How to spell processing? 1C, 2C? 1C. Okay, so there will be a little bit of processing. The word you may see sometimes is splicing. When we say the word splicing is this, we will take out and join things back together. So I'll remove all the red parts and I'll keep all the blue parts after I process it, the mRNA. Yeah. So that's what introns are. Introns are sequences that may not be so meaningful, and we need to take out. right? So that we are left with the meaningful sequences that will lead to amino acids that you want. Which beckons the question, why have introns in the first place? So troublesome. Okay? Certain instances, introns may contain amino acids that may come in useful. So let's say if I want to create one version of a protein that's like that. Okay? Let's say I'm creating a membrane protein. Okay? A membrane protein that can either bind to the surface of the membrane or a version of the protein that can float around. Okay? So what's interesting is this. I could create a version of the protein that is like that. Right? A certain sequence of amino acids, which results in a protein that floats around. Let's say I include the intron after splicing up. Now I may create a version of a protein that is now embedded onto the membrane itself. So, in other words, introns exons gives me another layer of control. I could take out certain introns, I could leave certain introns behind, and it will result in a totally different protein being translated. The totally different protein often is just changed a small little part. So for example, this protein over here maybe does not have any hydrophobic parts. This one over here has a hydrophobic portion, the red portion, perhaps because of this intron that's left behind. So it can now stick to the membrane. So that's the beauty of splicing. Splicing gives me that additional flexibility to modify my mRNA after it's been translated, to get different proteins at the end of the day. Next, where is tRNA synthesized? It's a great question. I don't know. But I think if we just Google now, we should be able to find it. Who posted this question? Maybe it's Dominic. So, where is tRNA synthesized? I'm not too sure where exactly it's synthesized or the enzyme that's involved. Okay, tRNA is called transfer RNA. Okay, if someone could Google and find out, and then just let us know. What is the purpose of the anticodon? Okay, just now in the video as we were watching, you observed that the tRNA comes with 3 codes at the bottom. Okay, so if I go back to the video over here, let's go back to
amino acids.
Okay, observe, this is the structure of tRNA. At the bottom, you see 3 bases sticking out. That's what we call the anticodon. The anticodon allows for the tRNA.
There are 20 different types of amino acid, with 3 bases at a time.
Allows the tRNA to bind to the mRNA strand. So for example, AGU, that's on the mRNA, because the tRNA has UCA, can complementary base pair to it and bring in the amino acid that we want. Right? So that's where the anticodon is, and that's its function. It's for recognition. Okay. How does the terminator work? Okay. Where is the terminator? Perhaps that's the first question. If there's a promoter to help you identify the beginning of transcription, there's also a sequence at the end. I can't remember the sequence. Okay, but the sequence is the terminator. The movie, right? Okay, that's the terminator, the robot one. Okay, so the terminator is sequence at the end. What does the terminator, how does it work? When RNA polymerase reaches the terminator, a whole slew of things will happen. A lot of proteins will start coming in to help it detach away from the terminator. Okay, so that's what's happening at the end. That's just a surface level understanding. Okay. Are different genes separated by start and stop codons? I think more accurate would be that it's separated first by promoters and terminators. Yeah? So different genes should have different promoters, terminators separating them. Then within the promoter and terminator, you will find the start and stop. Where does Trna get the amino acids from? Where do you think it gets it from? Now, where does Trna get amino acids from? It can get from the cytoplasm, but where did the amino acid in the cytoplasm come from? Yeah, the food you eat. Yeah? So you eat the food, proteins, you digest, then after amino acids, then it goes to your bloodstream, goes into your cells. These amino acids are now used for this process. Okay? So some amino acids in cells exist together with tRNA. Amino acids and tRNA exist together in the same cytoplasm region. You will find both in the region. If that answers the question. Next, how do you switch on a gene? Who asked this question? Okay, what do you mean by where do you read this? Okay, so how do you switch on a gene? When you go to the acid side, you'll find that beyond the promoter, there may be some other sequences. Okay. Beyond the promoter, sometimes you'll find other sequences that when RNA polymerase binds to will result in it going through the. Let me think. No, wrong. Okay, so how do you switch on a gene? One way is, you know how we package DNA to wrap around the stones? They have to wrap so many layers, right? Think about it, how is RNA polymerase going to get to transcribe it? Right? So one way to switch on a gene is to make sure that all these histones that are removed is uncoiled to a point where RNA polymerase can access it. So that's one way you can switch on a gene. Yeah. Sometimes on the promoter, there may be proteins bound onto it to block the RNA polymerase from even finding it. So one way to switch it on is to destroy the promoter or destroy the protein that's bound to the promoter. How does mRNA move out the nucleus and what drives the movement of mRNA? When you go to the university side, you'll find that there are proteins that help to transfer it out. Okay? What's the difference between DNA polymerase and RNA polymerase? Good question. What is the difference? Right, just based on what we've observed. I like the name itself even tells us the difference, right? So what's the difference between DNA polymerase and RNA polymerase? When was DNA polymerase used? DNA replication, yeah? So DNA polymerase used in DNA replication because we are creating new DNA. RNA polymerase is found in this process because we are creating RNA, mRNA. Okay? In fact, therefore, you could try to compare DNA replication, transcription, and translation. Oh my god.
Stop giving you ideas.
But I'm giving myself ideas. I already said the question, so this is
One of them.
I shall not say more. If I say no, it means something. If I say yes, it also means something, right? Somehow I'm being recorded. It's very scary. They're not really related, but why our body just use the T base instead of switching out for U base and transcription? Okay, that's interesting. Why create an mRNA when we have to swap out the T's to the U's? I think what your friend means is that I could have created a copy with T instead, right? Why create a copy with U? Okay, if I did T, then I'm creating a DNA replicate, a short DNA replicate. Okay. Did I say something? Oh, I don't know what I said.
Oh my god.
I couldn't rewind to understand what I said. Okay, but one of the reasons is actually if you have mRNA view, you can identify the mRNA. Yeah, you can identify the mRNA for degradation also. So the U kind of stands out, sticks out at the tongue. You see a sequence with U, and I could degrade it once I don't need it anymore. Yeah. Okay, why are there non-gene segments of a DNA? Okay. We say that it's non-gene segments because at the moment we don't quite know its function. There are a lot of sequences in our DNA that we don't quite know the functions yet. So we call them garbage DNA. For now, yeah, it really, the scientists call it garbage. But until we find out its true purpose, then we will know. Confirm got purpose, I cannot be recurring garbage now. How does the body regulate the number of introns they include? proteins. Everything boils down to proteins. There are enzymes and proteins that can help to regulate. There may be something happening in the body, some chemical reaction, some negative feedback that will affect what introns and exons are being put into place. Okay. Anything else? Are promoters and terminators bound permanently to the DNA strands? They are part of your DNA. They cannot be removed. Okay, so promoters and terminators are always there. Yeah? Can amino acids be found by themselves in cells, or are they always attached to tRNA? That's an interesting question. They can be found free floating. They can also be found attached to the tRNA. Okay. Last one. Oh no, that's the last one. Okay, so those are our questions. If you have any more questions you'd like to ask for transcription translation, I'll be glad to take them. Any more? If not, we're going to go into something more interesting. What if we modify your DNA? Okay, here is your mRNA transcribed from the template strand. I'd like you now to access activity 2 of your worksheet, and this is what you will find. You will find this simulation. Okay, this simulation is interesting because it allows you to modify one thing. Okay, when you're at this simulation, I'd like you to then click on Edit DNA, and you will then find the, what strand is this? You will find the coding strand. Okay? When you click Edit DNA, you will find the coding strand. This is the strand you will find. Okay? Now I'd like you to access the class activity. Yeah? There are 3 activities I'd like you to try. Okay? If I provide you the coding strand, I'd like you to see the impact of modifying the coding strand. First and foremost, I'd like you to see what the original protein looks like. Okay? Okay, so let's transcribe. I'm going to how to start this. Let's start this. Okay, this is a cell. We're going to go in. Okay, we're going to find a gene, and then we're going to transcribe it. Okay, RNA polymerase comes in, finds a promoter, binds the DNA, and then it begins transcription. Okay, let's transcribe. Pop, pop, pop. Okay, what is those? Okay, let's jump. Okay, transcription takes place until we reach the terminator. After transcription, you will then go through translation. Okay, where we bring in, oh no, I just kind of skip everything. Let's translate. You will see tRNAs being brought in, matching the anticodons with codons, and you'll find that the amino acid chain gets longer and longer. Right? You just form more peptide bonds, and you see the polypeptide growing or elongating. Eventually, this process will stop when we encounter a stop codon. So you look up for the stop codon, it's coming up. CUAG. Okay, when it reaches the stop codon, you detach, and this is the protein we will get. Your job now is to go through steps, I think 7 to 9. I'd like you to edit the DNA. Go directly into the coding strand and follow the instructions for steps. Which steps? I'd like you to skip all the way to step 7. Okay, so 7 onwards, there's substitution, insertion, deletion. Then last but not least, you can explore. You can do whatever you want to the coding strand and see what happens to the protein. As you edit the DNA, I'd like you to flip to the very last page also to collect the information. How did the sequence change? What's the new amino acid sequence? Okay, then we will invite some of you on stage to double check the sequence. Okay, so for now, you can work individually or work with a friend. And to edit the sequence, I'll play some music to keep us also occupied the ears. Okay, I'd like you to collect information along the way. Yeah? Write down how the DNA sequence has changed, as well as the new amino acid sequence that you get at the end of the day. of your activity, you should have filled up this final page. Right? Fill in the sequence. And you amend it, as well as the corresponding amino acids that you get. Before you edit for the second time, make sure you restore the original coding strand. Don't just edit your edited version. You may want to compare your results with your friends after you're done editing. Say again, before you do step 8, make sure you restore the original sequence. Don't edit over your edited sequence. it's not unique mutation. It's just by chance. Yeah, that's what we're okay. Yeah, give you around up to 9:30 for you to continue or finish up, alright, and then compare your sequence with your friends. You should get the same sequence for each of these steps. Sorry, let's go down this one. You have tried a few things by now: substitution, insertion, deletion. Which to you is the most serious? At least the most drastic change. Okay, I'd like to round up soon. Okay, I'm going to close off our activity. Okay. This is the data I collected when I played the game. I follow the steps up. Okay. Let's go first to your base pair substitution. This is something that you got. I've highlighted something, one of them in blue. That was the only change I observed. Was that the same thing for you? You have more change? Okay, so the first step was to substitute. Did you all get more change? By right, you shouldn't. Sorry? Oh, it's alanine. Ella, is it? Oops, okay. Is this better? Was that the only change? Okay, so for your information, I noticed I keep typing base pair. Right? Base pair substitution, base pair insertion. What do I mean by that? I mean this. Even though you were editing the coding strand, you're only swapping out like one of the nucleotides, right? But remember, this is a double-stranded DNA. So when you make a change to one of them, you're actually making a change to the bottom one also. So that's why we call it a base pair substitution. We substitute both at one go, right? But the one you only did for one strand. That was the coding strand. Okay. All right. So if you did exactly what the instructions told you to, the only change you have observed is here, right? Okay. I'm going to now introduce you to some terms, some forms of mutations. Okay. If you see that the resulting amino acid is very different, it's totally different from the original amino acid, right? You see codon 2, codon 2, the amino acid is totally different. We call it a missense mutation. M-I-S-S-E-N-S-E. Missense, right? It's a missense mutation. If an amino acid becomes totally different from the original amino acid in its place. Okay, so there is a missense mutation. Originally it was a proline, now it's an alanine. We call it a missense mutation. Okay, you could highlight that particular example as a missense mutation, codon 2. For those of you that did the insertion, okay, everything I highlighted in blue changed. Okay, I say again, everything I highlighted in blue changed. In other words, as a result of this base pair insertion, I get many missense mutations along the entire amino acid. Many missense mutations. But I'd like to draw attention to two amino acids. Did you get two leucines, CTT and CTT? Okay, I'd like you to highlight those. Compare it to the original sequence. At the very same spot, the amino acid is the same. Right? If you compare back to the original sequence, actually at these two codons highlighted green, it was still leucine. Right? We still get leucine despite the drastic changes. But what's the difference this time? The code is slightly different. If we compare to the very top, Leucine was created from a TTG. Water is created from a CTT. But we still get leucine. So on the surface, nothing changed. But underlying, something changed. We refer to such mutations as silent mutations. Because you can't tell anything changed on the surface. But underlying, the genetic code has changed. Okay, so that's a silent mutation. Okay, so two kinds of mutations so far. Missense, silent. When you did the base pair insertion, notice something drastic occurred. What was that drastic change? A stop codon just popped up somewhere, right? If a stop codon suddenly appears as a result of either a substitution, insertion, or deletion, we call that a nonsense mutation. Okay? This is nonsense. How dare you do this to me? Haven't complete my order already, you go and cut the receipt. Okay? So if you encounter a stop codon that suddenly pops up, that's a nonsense mutation. But you can also see, again, lots of missense mutations. Insertions and deletions are really causes a lot of change. Then many of you explore. Do you get very drastic changes in your exploration? Three kinds of mutations that can occur at the genetic level: missense, silent, and nonsense. Hope this makes sense to you. Okay, so if you ever hear the word mutation again, as a biologist, understand that when we say the word mutation, we're talking about changes to your genes. Right? Yes, you see, like, you know, you watch movies like X-Men, right? When they mutate, right, it's really like claws come out from the hands, right? Yeah? That is a superficial understanding of mutation. Now, when we talk about mutations, we need to have a genetic change. So there was a genetic change that resulted in Wolverine having all those claws. Okay, so mutations. Hey, this is genetic mutation. To end off, I'd just like you to show. I'd like to show you another kind of mutation on the chromosomal level. Chromosomal mutation. This is us making small little edits to the recipe. I just change out one word, without one word. Then your sentence doesn't make sense. But what if I make changes to the entire chromosome? Right? I'd like to show you a final video to end off today. Chromosomal mutation. What is this?
In a sheep shroud, innovating the world of education.
First kind of chromosomal mutation. Sometimes during DNA replication, you may end up replicating one part, one whole segment, multiple times. Now we're not even talking about small little edits. We're talking about maybe thousands of genes in one segment being replicated twice, right? And put within the same chromosome. That is a form of chromosomal mutation. We call this duplication. Another form of chromosomal mutation, maybe an entire segment is totally removed accidentally during replication. Maybe replication we skip some parts. Understand that this is not just one gene affected, it can be thousands of genes in that segment. So this is another form of chromosomal mutation. Translocation. Sometimes you have two chromosomes that are not even homologous. They end up swapping parts. It's very drastic. It's not like homologous chromosomes exchanging alleles. You are totally changing genes from one position to another. It can affect very on a surface level what is being shown. Inversion. Sometimes the segment of the DNA is totally inverted, and that will mess up how you read the entire sequence. All the genes in that region will be read in a different way. You'll get proteins that totally make no sense. Polyploidy. Sometimes all your anaphase processes may not occur well. Okay, so let's rewatch that a bit. Okay, look at meiosis on the right side. Let's look at faulty meiosis. See what happens in a faulty meiosis? One of the cells didn't even go through anaphase. And that's something that can happen within our cells. A lot of times we talk about mitosis and meiosis in a very perfect sense of the word. But actually, a lot of times mistakes can happen. What if the kinetochore doesn't attach to the microtubule? Then the chromosomes aren't even pulled apart. Right? That can happen. And if that happens, you may get gametes that end up fusing with each other. You may end up with a triploid organism at the end of the day. Right? So, two ways chromosomal mutations can occur. One, because of faulty cell division processes. Another, during DNA replication, big mistakes happen. Yeah? So, two ways in which chromosomal mutations can happen. For genetic mutation, we have seen three different processes that can take place. Right? One is substitution, one is insertion, one is a deletion. And they'll cause different kinds of mutations depending on where you perform these things. But not to worry, notice we are not constantly mutating because our cells have these checks in place. Okay, almost done. Okay, so that's all for today. Next time around I'll see you. We'll just do a quick recap of this chapter, and then we'll move on to the next one. I think the next lesson I see you should be our quiz, type for human reproach. Okay, so you will have RE 6.1. This will be due on the 26th of August. Okay, thank you. Wendy, do you help me stop the video? Okay, that's all for today. I'll see you again.